Okay, good morning. So today we are going to discuss about the topic resins. Okay. So previously we have discussed about the secondary metabolites like alkaloids, glycosides, flavonoids. So the topic we are discussing today is the drugs that comes under category of resins. So about resins you have previously studied in 4th semester, right? Resins are secondary metabolites, and here the drugs that comes under category of resins are ginger, asafoetida, myrrh, colophony, guggul. Okay, so we will be discussing in detail about ginger. So I think you know about ginger. Ginger is a common drug that is used for daily purposes, in our daily routines, for medicinal purposes as well as for edible purposes, right? In food, spice productions, etc. So in pharmacognosy, we are in detail studying about the features, the morphological features of the drug, the microscopic features of the drug, the biological source, various chemical constituents present in the drug, and then those constituents that is responsible for the therapeutic activity. Okay, so the basic outcome of this topic is to study in detail about the ginger. Not only ginger, all drugs. I think you know most of the drugs that comes under pharmacognosy is a familiar drug for you. But we are not studying about the morphological features or transfer sections, right? So in pharmacognosy, the basic course outcome is to study in detail about the drug. So shall we start? Okay, you can write down the lecture notes. So first one is the biological source. Okay, so when note downing the biological source, you have to note down the plant part, the biological name as well as the family. So in case of ginger, the biological part is roots or rhizomes. So it is mainly obtained from roots or rhizomes, that is mainly obtained from Zingiber officinale belonging to Zingiberaceae family. The chemical constituent, major chemical constituent present in ginger is the volatile oil. The ginger oil consists of monoterpene hydrocarbons, sesquiterpene hydrocarbons, oxygenated monoterpenes and oxygenated sesquiterpenes. In addition to it, it also consists of zingiberine, which is an important constituent, gingerol, another important constituent, and gingerol. Zingiberine, gingerol and gingerol. These are the major constituents present inside the ginger that mainly provide the therapeutic activity. Clear? So the basic test, chemical test that can be performed to identify the presence of resins in ginger is 1 gram of ginger is taken, mixed with 5 ml of acetic acid. Shake the mixture well for 15 minutes and then filter it. To the filtrate, add ammonium oxalate and we can see turbidity. Okay, to identify the resins present inside the ginger, we will take the sample along with 5 ml acetic acid and then we will be shaking the mixture for 15 minutes, filter. To the filtrate, add ammonium oxalate and we will be seeing slight turbidity. So if turbidity is present, it indicates the presence of resins in ginger. Okay, this is the specific test for identification of resins in ginger. Next is cultivation and collection. So when we are discussing about cultivation and collection, first we have to note down the conditions required for cultivation of ginger. So the first condition is humid climate, where the rainfall should be 80 centimeter annually. 80 centimeter rainfall annually with humid climate, heavy irrigation, and the plant part that we are using for cultivation is bud. So the bud will be transplanted into the field at the month of March or April, and then the plant will be growing, and once the leaf turns to yellow color, from green to yellow color, we will be harvesting the plant. Okay, so the harvesting will be done after 6 months, that is almost in January or December, by digging method. Since the plant part is rhizomes, we have to completely dig out the plant to obtain the rhizomes, right? So we will be digging out it after 6 months of cultivation, and then after that we will be washing it, drying it, which is the pure ginger that is obtained. So that is the ginger that is available in the market. Clear? So in cultivation, hot humid climate, 80 centimeter rainfall, heavy irrigation, and the cultivation is done during the month of March or April, where bud is the plant part used. Okay, so once the plant turned from green to yellow, once the leaf turned from green to yellow in color, we will be harvesting it, complete digging out method, and then the collected rhizomes will be washed, dried. Okay, Is it clear? Cultivation. So the cultivation will be started on March or April, and after 6 months harvesting will be done. So next, the macroscopic features of ginger. So here we are mainly studying about the various features that can be identified by various senses. Okay, first one is color, that can be identified by eyes, right? So the color of ginger, outer color is buff color. Next is the odor, which is aromatic odor. The taste, pungent taste. Fracture, short and fibrous. Coming to the shape of the ginger, we have to mainly mention that the lateral surface of ginger is flat. Okay, and it also consists of several branches as seen in the images. This is the macroscopic identification of ginger. Next is the microscopic features. So when coming into the diagram, we can see here the first outermost layer is cork. Okay, cork is the outermost layer, which is mainly made up of irregular cells, irregular arranged cells. The cork is divided into two portions, outer cork and inner cork. The feature of outer cork is dark brown in color, and the inner cork, it is colorless, followed by cortex region. So the cortex region is mainly made up of parenchymatous tissues. It also consists of starch grains, oleoresins, as well as a individual layer of endodermis. Clear? So the first one is cork. Cork consists of outer cork and inner cork, followed by cortex region. Cortex region is made up of parenchymatous tissues, and it consists of oleoresins, starch grains, and as individual layer of endodermis. The last layer is about vascular bundles. So it consists of xylem as well as phloem. Okay, in case of tears of ginger, dorsal surface consists of only xylem vessels. That's why the image is marked with xylem. Okay, phloem will be present in the ventral surface. Clear? Microscopic features: cork, cortex region, and vascular bundles. Cork consists of outer cork and inner cork. Cortex consists of starch grains, oleoresins, and a layer of endodermis, followed by vascular bundles. That is the microscopic features. Next is several adulterants and substitutes that can be used instead of original ginger. Okay, so the first one is cork ginger. So you have to mark the specific features. So first one is cork ginger. It is mainly used for fresh consumption as well as medicinal purposes. So the deviation from original ginger is that the rhizomes will be larger in size, smoother skin. Okay, the rhizomes will be larger in size, smoother skin, light yellow to pale yellow brown color. The color of original ginger is buff color, right? So here the color of cork ginger will be light yellow to pale brown in color. That is the specific feature. With this feature, we can identify whether the ginger is original ginger or adulterated ginger. Clear? So the second one is Nigerian ginger. It is mainly used for spice productions, not for medicinal purposes. The features is rhizomes are smaller and thicker rhizomes with compact structure. Original ginger consists of lateral surface with flat surfaces. Here it consists of compact structure, and the color is light brown to dark brown in color. The third one is Jamaican ginger. It is mainly used for culinary purposes and in pharmaceutical industry. Culinary purposes means for food production. Okay, so the specific feature is it consists of smaller rhizomes, but the difference from Nigerian ginger is that in Nigerian ginger it consists of compact structure, and in case of Jamaican ginger it consists of straight structure. Okay, and the color is golden brown in color. Next one is Chinese ginger. It also consists of smaller rhizomes, which is mainly used for cooking purposes in fresh form. Okay, in fresh form we will be using Chinese ginger. The color is light brown to yellow brown in color, compact and straight is the feature compared to original ginger. So total there are 4 adulterants and substitute, that is Chinese ginger, Jamaican ginger, Nigerian ginger and cork ginger. These are the substitutes that can be used instead of original ginger. Next is the therapeutic activity or therapeutic uses of ginger. It is mainly used as stomachic, aromatic purposes, to lower the cholesterol, expectorant and flavoring agent. These are the specific uses of ginger: expectorant, flavoring agent, carbonatives, aromatic, stomachic, and always also to lower the cholesterol level. Coming to the commercial applications, as I said, it is used in culinary industry, that is food industry, pharmaceutical industry, for aromatherapy, for massage therapy. Okay. Additional to pharmaceutical purposes, it is also used in food industries, massage therapy, and aromatherapy. So summarizing the topic, we have discussed about the biological source. Initially, we have started with the biological source of ginger, followed by the various constituents present in ginger, and then we have discussed about the how to cultivate and collect the plant part, and then the macroscopic and microscopic features along with adulterants and substitutes, and the specific therapeutic uses and commercial uses. Okay, is it clear?